So I realized that I'm the only one standing between you and the imaginary, grandiose grand finale conference closing dinner that most likely Nikolai envisioned at the end of the conference, and most likely he selected nice restaurants, fancy restaurants to host us, legal seafood or what have you. Unfortunately, it's not the case, and most likely we can still hope for some magic and some box dinners to show up in our doorsteps with lobsters, champagne, and oysters. But if not, then hopefully next year we'll see each other in person. So I wonder if you can see my, if I can start sharing. Oh,
we
great.
can see the slides. They're not full
okay.
screen, but we can, now they're full screen.
Okay, great, thank you. So let me start. So I was thinking about presenting several topics that we are actively working in my lab: sample preparation, different modalities of separation coupled to mass spectrometry, LCC. But at the end, I decided to narrow down the subject of this presentation to coupling ultra-low flow rate separation, namely LC, namely using monolithic columns, and coupling the separation to mass spectrometry using the FAIMS interface, which we recently, about less than a year, probably 6-7 months ago, we got our hands on, and it was a nice toy to play. So we decided to check whether the technology is compatible to ultra-low flow rate separations and analysis of limited samples. I would like to start with the acknowledgement slide and thank my lab members. And as you see, during the last few months, we got used to Zoom meetings, and we had some fun also over Zoom, even cocktail hours. And so I'll get to this slide later, but I greatly appreciate energy, hard work, and dedication of the lab members into the ongoing research. So mainly I'll be talking about nanoflow and mainly about ultra-low flow coupled to mass spectrometry. And we evaluated the FAIMS Pro interface introduced probably last year by Therma. And now we have a couple of them, access to a couple of them around in the lab. And then just 2 samples, a model HeLa protein digest standard that everybody is using, and monocyte cells, cultured cells that we initially got from Nikolai's lab. And I would like to, here, let me move things around. To acknowledge Michal Gregush, mainly I'll be presenting his work. So he has been working very hard on different types of ultra-low flow separations and coupling them to mass-spec. Also James Costas, he has been very instrumental in developing and advancing sample preparation techniques. Somak, who helped with data analysis, and Sue and Kendall. So Sue was just extremely helpful with the FAIMS Pro interface. She's a pro in this field, and it was great to working with her on the project. And Kendall, I'll show just a few slides from her recent work. So basically, as an overview, we have several modalities, several types of columns, either bead-packed columns shown on the top, and we play with different diameters, different flow rates, open tubular or porous open tubular columns like shown in the middle that we are actively developing and using in the lab. And also those porous monolithic columns. It's a sponge, porous sponge within the capillary, and we typically use polymeric monoliths to polymerize either using UV light or temperature in capillaries. And there are benefits of one versus another type of separation modalities. But today I'll mostly show data acquired using PSDVB monolithic columns, which are shown on the bottom of this slide. So the setup is pretty simple. We are using regular Dionix LC systems, normal autosamplers, and with minor modifications. We just put a plug here and a T, which allows us to load our monolithic columns, which are 20 micron ID at low flow rates. Directly on the column without loading, without using any SPE columns, any trapping columns. And of course, there is a trade-off in time. So it takes about 60 minutes, typically 60 to plus minutes, depending on the sample volume to load the sample on the column. Then we use this line as a splitting capillary and run our normal ultra-low flow LC separations at the desired flow rate. In this work, I'll be showing data acquired using 12 nanoliters per minute, and the columns were 35 centimeter long, and the gradients were always 60 minutes, even though we also use 60 minutes to load the sample. So that's the setup. Previously, our group and other groups showed benefits of ultra-low flow rates. Here's an illustration. So we are getting signal intensity increased in mass spectrometry. So we are getting a lower ion suppression, better transfer of ions into the mass spectrometer. So the overall gains have been shown before, but of course there are still difficulties and challenges using these columns. So we are greatly dependent on the dead volumes, connections. Optimization takes a while at these flow rates. There are no pumps available for direct elution and loading at such flow rates. So there are issues. Some of them are listed on the top. Applying spray voltage, spraying from these columns might not be trivial. And you saw a nice talk by Ryan Kelly yesterday. He pointed out their solutions to this. So anyway, we decided to use these columns since they allow us to load directly the exact amount or the total amount from a very small limited sample on the column and analyze it with FAIMS or without FAIMS. So that's pretty much the setup, and the elements of the workflow we were using Orbitrap Fusion Lumos as a mass spectrometer, and either with or without the FAIMS interface. So as you know, the FAIMS stands for high field asymmetric waveform ion mobility spectrometry, and it separates ion dependent on their mobility in the asymmetric field. This particular technology available from Therma does not provide very high resolution. Nevertheless, the hope was that we can get rid of single charged ions, we can decrease the noise level, and improve our detection sensitivity. So the main manipulation with the parameters was on the side of setting different compensation voltages for the FAIMS interface. So when we got access to the interface and the FAIMS technology, what we knew, that's fine. Yes, ultra-low flow LC helps increase the signal intensity in mass spectrometry. FAIMS technology was already shown to improve the signal, improve the depth of proteomic profiling. At that time, the published examples, published reports were dealing mostly with higher flow rates from 100 to 400 nanoliters per minute and significantly larger sample amounts. So recently, again, Ryan Kelly's group published nanogram level samples with the FAIMS. And I'm glad to see that we found very similar trends. So the question was whether, and actually the previous report showed that there is some loss in signal and incomplete ion transmission through the FAIMS interface. And the question was whether it would hurt us with ultra-low flow rates, whether the ultra-low flow rate separation can be coupled to mass spectrometry using the FAIMS technology. And then can the FAIMS technology be applicable to limited samples and potentially single cell analysis? So that's what we were trying to find out using two types of samples that I already mentioned. So definitely with sub 20 nanoliters per minute flow rate and very small sample amounts, we don't expect to see high ion flux going into the mass spectrometer, and any losses can be harmful. So back to the setup again, I discussed the left part, the monolithic columns, that's how they look, the LC system, and the two types of samples are shown here. The only difference was with and without FAIMS interface, and the hope was to remove singly charged ions and improve signal-to-noise ratio. So with helodigests, we played quite a bit, and we changed the number of compensation voltages. So we started from published recommended settings with a single compensation voltage. Somehow it didn't work out for us. It didn't help. So then we moved to 2, 3, 4 compensation voltages. They're shown in the table, and different time per CV, per compensation voltage. So which defined the cycle time in our data acquisition mode. So, and the total cycle time is shown on the right, in the right column. And also we optimized the control experiments and brought them to the level where we got highest numbers of proteins detected using 60-minute long gradient. So at the end, we selected a FAIMS method with 4 CVs and 1.5 seconds per CV, and used it in all subsequent experiments. So the main design is when we flip between different CVs, let's say 4 CVs, we acquire full scan MS1 data using the same CV, and then go through data-dependent MS2 scans using the same CV, even though if you check the raw data, there is a, maybe I should, there is a kind of a switch, a jump between one level of CV to another due to most likely to parallelization in data acquisition on the LUMAS. So the first thing that we explored was whether indeed we are getting similar signal intensity and whether we are gaining in the signal-to-noise ratio. So if you look at the left set of panels, A, B, so the base ion chromatogram signal is pretty similar for FAIMS, no FAIMS experiments, maybe some fluctuations there. And remember that in the FAIMS experiments, we keep switching from one CV to another. The signal is a little bit jerky, it goes up and down. But nonetheless, the overall intensity is fine. Then if you look at any empty region, retention time range on the LC-MS chromatogram, in this case, I'm showing an example from the end of the gradient after all the peaks are eluted and the column got equilibrated. We did the same thing running blanks in the middle of the gradient. And what we observed that the signal changes about 3 orders of magnitude, just the baseline signal. And that was encouraging. So, and it was quite robust. And if we looked at the corresponding spectra, MS1 spectra here, you see that in the case of the FAIMS, this spectrum is pretty sticky, no data, so the thermal software doesn't allow us probably to go down to the noise level. And in the no FAIMS control experiments, we see our regular suspects, 445 and other polysiloxane ions present in all spectra, and also their adducts, and so additional peaks. The complexity of the spectral data goes up. If we check the whole run using ion density maps as shown on this slide, in the control experiments, you see those streaks which correspond to singly charged ions, polysiloxane ions, which are mostly removed with the FAIMS in the FAIMS experiments. And if you look at the data acquired at individual CVs, 4 examples are shown here. Also, the data looks significantly cleaner. Even the intensity is approximately the same. So, and then it's, as Chris discussed a couple of talks before, the benefits are clear here. So we don't transfer those single charged ions of high intensity in the orbit trap, in the mass spectrometer. And so there are multiple benefits that can be expected out of that, including MORZ changes, ion coalescence, and so on. So if we looked at base ion chromatograms extracted at each of the 4 CVs, compensation voltages that we used, so they do not look alike. That's encouraging. It means that there is some complementarity between different voltage settings. And then if we check the protein IDs for 1 nanogram of helium, in this case, unique and total. So we see that in each compensation voltage, we are getting a unique sets of PSMs, peptides, and potentially proteins. So that was helpful to see for such low levels as 1 nanogram helium. We are not at the level of single cell, of course, here, but somewhat close. This slide shows our optimization steps where we played with different CV settings and different time allocated per each CV cycle. So at the end, if we select, we compare control with optimized conditions, we're getting about plus 40% IDs for proteins and significantly more for peptides. At the level of 1 nanogram, similar gains, but maybe less pronounced gains are observed for 100 nanogram of helium. So that proved that, yes, the technology can be efficiently used for ultra-low LC, ultra-low flow LC, and also for small sample amounts. In terms of the reproducibility in between the runs, we used MaxQuant in this case. We extracted LFQ data, compared FAIMS replicates, control replicates, and FAIMS to control. nice correlations, and you see diagonal, the majority of measurements, LFQ measurements are laying on diagonal for each control versus control comparison, FAIMS and versus FAIMS comparison, but there is a little bit of a shift when we look at the comparison between control and FAIMS. And that shift can be better seen in this slide. If we plot LFQ data, so the label frequent mutation data, again for 1 nanogram HeLa and 1 hour gradient. So in this case, we show only proteins matched between controls and FAIMS, and with IDs having at least 2 quantitative measurements, I think. So the missing the IDs with multiple missing data points, LFQ data points, were filtered out. I don't remember exactly what were the settings, so we got down to about what 1,007 or 800 proteins here. As you see that the quantitative measurements for FAIMS are LFQs are a little bit lower, even though we are able to detect more protein IDs. But at the same time, the dynamic range is slightly wider, slightly broader. So, and the LFQs, all data shown for 3 replicates, FAIMS versus control, again, show the shift. So lower intensity for the FAIMS data, slightly higher intensity for the control. Nevertheless, we are gaining the depth in proteomic profiling using the FAIMS interface. So what we also hope to see is the ability of the interface to remove effectively singly charged ions, and we do see, if you compare the blue bars, we see that with the FAIMS technology, we were able to decrease the number of singly charged ions if we compare to the control with individual CVs, and if we combine them together. And even though the number of features, singly charged features, may be like what, 2-fold different between the FAIMS and control, the intensity was significantly lower. They were barely detected, and that resulted in cleaner data. About 2-fold more multiply charged ions were observed when we used FAIMS. So also, if we look at the sequence coverage data for proteins, the same filtered about 1,500 proteins, we see that on average we are getting better sequence coverage for proteins versus the control experiments without FAIMS. Here, matched data, matched proteins, and the corresponding sequence coverage for FAIMS experiments in orange, and in blue dots control experiments. So that was helpful. And also, if we check ion interference for MS1 isolation, expectedly we got good gains. So in comparison to control, the on average we were getting significantly lower ion isolation interference, and that's we were using ion trap data. We're selecting precursors of 1.6 m/z units. So for TMT, it should be significantly better when we use orbit trap data. And we don't have I don't have data here for TMT labeled limited samples, but we already ran some experiments for bulk samples, and we see clear benefits of the FAIMS. On this slide, I pretty much summarized the identification results, control versus FAIMS, and we are getting about 28, close to 30% gain in protein identification. So protein groups, larger gains in peptides. So, and this is all for 12 nanoliter per minute flow rate and 1 nanogram of HeLa. Similar gains were shown for 50 nanograms, 100 nanograms, and so on. Slightly less significant gains, but for 1 nanogram it was pretty good. So let me see. So then we switched from the control experiment to the U cells, U937 cells. And in this case, we used pretty rudimentary approach. We published quite a few years ago on processing samples on micro SPE tips, like stage tips, or similar to stage tips. So we improved a little bit the protocol. We played with the protocol. It works really well for samples of the amounts like low 1,000, low in a few hundred cells, up to 10,000 cells works well. And will not work for single cell processing, obviously, because the surface area is still quite large. So James Costas was involved in it, and he would be happy to share more details, but as you see, he's on mute here on this slide. So maybe at some point later, we are trying to finish up that simple story with him. So how we designed the experiments, in this case, I'm showing 3 levels of cell numbers. So we took either approximately 100 or approximately 1,000 cells or 10,000 cells and processed them on a membrane, actually in the membrane, which we used as a microreactor, and then eluted with a small volume and injected 1/10 of each sample. So roughly for the smallest sample amount, cell number, without considering any losses, we are injecting the sample equivalent to 10 cells approximately out of approximately 100 cells. So definitely it's not like 10 cells selected and then picked and processed, but we are trying to do this proof of principle experiments before getting to the single cell experiments. So pretty much the same story if we compare FAIMS and control experiments. So those streaks for singly charged polysiloxanes are gone. So also the signal for trypsin autolysis peaks is quite a bit removed. We don't see those adducts here in the FAIMS experiments. So the data looks cleaner using the same intensity on this comparison. So the numbers are not as great as for 1 nanogram of HeLa because of the losses and suboptimal sample prep, but the gains are similar, FAIMS versus control. And it is pretty much similar for different levels of cells processed. So if you processed 100 cells and injected 10, 1/10, we saw about 40% gain, and then about 20% for 100 cells when 1,000 of cells was processed, and so on. So, and for peptides, the results are shown on the bottom. So I think, yeah, here is a summary of the numbers, plus 52% unique protein groups and plus 78% unique peptide groups. That's quite similar to what we saw for HeLa, and that's exciting that the technology can be applied for such small sample amounts. So to summarize this part and the main part of my talk, one of course comment is that the FAIMS technology is compatible with ultra low flow, and we tried it already with lower flow rates. So it seems that it can work robustly. It can improve the sensitivity, the depth of profiling in proteomics experiments of small samples. So hopefully it will help with single cell proteomics as well. So the noise level was substantially decreased, like 3 orders of magnitude. That helped. I think we got quite good numbers for 1 nanogram of HeLa using 1 hour gradient. So around 2,300 proteins, which I think favorably compared to what was published before for the same HeLa standard. And I think even longer gradients were used and slightly higher sample amounts. So for U cells, got more modest results, but also the method is working, and the sample preparation works better for somewhat larger samples amount, a few thousand cells. And that shows the potential compatibility with the single cell analysis using both ultra low flow and FAIMS. So I decided not to show much on our ongoing CE-MS efforts. We're working quite actively in the analysis of glycans, released N glycans. By CE-MS, we are profiling them, and we're using very tiny volumes of biological fluids, plasma, and isolates from plasma. I presented some of that data last year, but some of the recent data generated by Kendall is shown here. We also compared FAIMS versus no FAIMS setup. In this case, we did not use ultra low flow LC-MS, but we used ultra low flow C-MS, and the SciEx instrument was used in this case. Quite similar gains, maybe slightly less we are gaining using different FAIMS settings and different number of CVs shown here. The similar data is acquired for peptides. For PSMs, it's totally different. So for the control experiments, we have tons and tons of singly charged ions there, ADAX and so on, possibly because of the buffers. So on average with FAIMS, we were getting at slightly larger sample amount, 8.8 nanograms in this case of the same HeLa standard. We were getting 12, 13% gains. That's how the separation looks in CE. And what's interesting in CE separation, that based on the differences in charge and hydrodynamic volume, we are able to pull out and enrich in a specific cluster phosphopeptides. So, and they're shown in this plot in blue. So pretty much they migrate late in the game, and we are trying to spread the separation to increase the depth. Of course, we are not able to spread it as well as we can do it in LC-MS based on hydrophobicity. But the advantage here that we are separating phosphopeptides without using any enrichment in this case. So, and then when we apply FAIMS and play with different numbers of CVs, we are able to increase the number of phosphopeptide identifications using the FAIMS interface as well. So that might be a unique feature of CE-MS, even though we are not yet reaching the depth of proteomic profiling with CE-MS, but maybe we can get additional information on the phosphorylation status side, as well as glycosylation, which we worked out quite well. So the same observations were made that we are improving the signal to noise, decreasing the level of noise. You see that the streaks of singly charged ions are gone. That results in some gains. It's not day and night gains, but at least plus 10%, plus 15 maybe, potentially more. On the peptide level, it will be more. Can be achieved. On that, I wanted to bring a few points to the discussion. As many of you remember, 10 years ago, 15 years ago, maybe 20 years ago, non-LC-MS, it was like black magic. Some people were packing columns, pioneers in the field like John Yates. They were packing multiphasic columns. So in many labs, non-LC worked, and some labs non-LC did not work. During the last decade, non-LC is present in pretty much any metabolomics, proteomics, lipidomics lab. All our mass spectrometers are equipped with quite robust, reliable LC systems. There are still room for improvements for LC systems, for autosamplers, for LC columns, but nevertheless, it's been a huge progress made during the last decade. So with ultra low flow columns, 20 micron, 10 micron ID columns, similar progress can be made during this ongoing decade. And this way we can enable new exciting applications, biological, clinical applications, single cell proteomics can benefit from that. So I hope that jointly as a community and together with instrument vendors, we can make that push and bring the separations to a different level. Mass spectrometers improved quite a bit during the last 10, 15 years. So, and then interfacing, including something like FAIMS interface and other improvements, even simple pool tips, coated, non-coated, how we couple them together. Again, I'm referring to some of the slides shown yesterday by Ryan Kelly, union or integrated column, what have you. So more progress is needed to make the analysis of limited samples and single cell more robust, more reproducible, more informative, and many problems, interesting challenges and problems can be solved this way. So, single cell proteomics can be also made robust, reproducible, and widespread. And it was very amazing to see the progress in the field made during last year and during the last 2 years. The quality of work, the results shown excellent progress. So with that, again, I'm coming back to the acknowledgement slide. I would like to thank lab members and collaborators like Sue. So, and this time around, I would like to also thank collaborators from Therma and SciEx, Nikolai personally and his lab members for helpful discussion, for providing some of the cells, and of course, the funding agencies for funding my research. And before finishing the talk, I would like to invite you to the next MSB 2021 conference that I am organizing together with co-chairs Kim and Jared. So originally we were planning to have this meeting in April of 2021. So because of the current circumstances, we moved the dates to July. I hope it is safer. It will be safer at that time. And I hope that we'll have an exciting in-person meeting. I invite everyone to this meeting. Usually it's a lot of fun. It's a small Gordon conference style meeting, rather small and interesting. So with that, thank you very much for your attention. Thanks again, the organizers, for inviting